All right, hello there. My name is Catherine Best, but I mainly go by Katie. That's what most people call me. I am a biology major with a chemistry minor here at Oglethorpe on the pre-med track, like I'm sure most of you are. Today, I wanted to share with you all some interests I had in the field of biology on the origins of eukaryotic cells. Evolution has always been a fascination for me. So when trying to come up with a research topic, I was immediately drawn to a specific article that came out of Japan in 2020 that I'll be breaking down for you all. The title of my presentation is a little lengthy, so bear with me. But molecular analysis of the Asgardian Archaea MK-D1 provides insight into early eukaryotic evolution. So today, I will be walking you all through the idea of eukaryogenesis and the standing theories of eukaryotic evolution, while also providing some pretty cutting edge research on how some of these theories have recently been altered, and then explain what the next steps in this type of research are. But before we get into the fun stuff, it is of the utmost importance for some background information. So we'll start with eukaryogenesis. There's kind of this really large question that keeps even the brightest of biologists up at night. And that is like, what is the origin of eukaryotes? I'm sure most of you are aware what a eukaryotic cell is, but just in case, the domain eukarya is one that houses organisms whose cells have a nucleus enclosed within a nuclear envelope. And they differ from the other two domains, bacteria and archaea, due to this membrane-bound organelle and the presence of a nucleus. Now, the questions surrounding the evolution of eukaryotes arise mainly because after reviewing the idea of evolution, which is briefly just the theory of organisms evolving due to seemingly random mutations that are beneficial to the host's survival, like natural selection. But I mean, after reviewing this, it doesn't really line up with the progression of an extremely complex cell from that of a prokaryotic cell, which leads us to two theories, which I will be reviewing for you guys today. First, we have the endosymbiotic theory, which I'll be talking about very briefly. And then after we kind of get into some of the research, we'll actually go over the newer one, known as the E-cube theory. So, endosymbiosis. This was originally proposed by Lynn Margulis in the late 1960s that further developed well into the 1970s. And this endosymbiotic theory was revolutionary. In her book, The Origin of Eukaryotic Cells, Lynn provided data supporting her theory behind this evolution. She claimed that a major organization event in the history of life probably involved the merging of two or more lineages through something known as symbiosis. And after observing the structure of organelles, such as like mitochondria and chloroplasts, she realized that these organelles resembled the structure and size of bacterial cells. So this, along with the fact that they both contained their own DNA, it was definitely enough to lead to research that would eventually imprint her name into the history books. Her theory was that mitochondria and chloroplasts are actually descendants of once free-living bacterial species. And more specifically, she believed that the mitochondria was from an endosymbiotic bacterium capable of aerobic respiration. And I kind of put that in little asterisks for you verbally, but through the process of phagocytosis, which is also important to remember, and that chloroplasts were from free-living cyanobacteria engulfed by a protozoan and reduced through metabolic slavery, in her words, not mine. But her genetic evidence was provided by the fact that both of these organelles were composed of 70S ribosomes, which are consistent with both bacteria and archaea. And it became clear that symbiotic events had a profound impact on the organization and complexity of many, many life forms. However, these revolutionary findings were met with so much criticism, and it's not really hard to believe that a lot of it revolved around the fact that, I mean, she was a woman when she was in a field dominated by men. But those who were able to get around the idea of her being a woman couldn't get around the idea that it was more of an unnatural sense of biology rather than the natural one that they were used to talking about. And by that, I mean, like the biology that had been proposed before this theory was one that was about dominance and harsh natural selection and mutations. And then the one that she proposed was kind of like in light of engulfment and immersion and a mutual change. And I mean, the notion that we are basically children of bacteria was just outlandish at the time. And this went directly against the Darwinistic idea that I mentioned before. Now, I mean, just as mankind's grasp of science is ever changing, there have been recent studies that suggest instead of it being bacterial cells, like Lynn thought, that engulfed one another, it was actually archaea that plays a larger role in the evolution of eukaryotes. So to kind of provide some evidence behind that theory, I have included 3 research papers that I'll just kind of briefly go through. We have the first one by Lopez Garcia that was published in 2015, which brings to light the controversy of a mitochondrial host. He mentioned how it is known that the last eukaryotic common ancestor was complex and that endosymbiosis definitely played a crucial role in the eukaryogenesis, at least via the acquisition of a alpha proteobacterial ancestor or mitochondria. But the nature of how it happened is controversial to him because in some of his discoveries that he outlines in this research paper, there is an archaeal lineage that shows it is phylogenetically closer to eukaryotes, which actually reinforces the models of a eukaryote-derived host rather than a bacterial-derived host. The second research paper that also came out in 2015, I have here from Martin, where it brings up the fact that most of these theories that Lynn provides and others provide actually fails to take into account the anaerobic eukaryotes, which is why I kind of put the verbal asterisks of the fact that she was talking about anaerobic eukaryotes. And then finally, we have the 2017 study, which is kind of the cherry on top for the idea of an archaea derived host. So they proposed an Asgard archaea that illuminates the origin of eukaryotic cellular complexity, because that's kind of where most people can't really get on the engulfment idea, because it's so hard to come from an extremely complex molecule from a simple molecule. But this Asgardian superphylum that they talk about, which is kind of like a gourd of uncultivated archaea, is related to another archaeal phylum known as Lochia archaeota, which I just call Loki for short. So that's how I'll refer to it. But Loki shares an astonishing amount of eukaryotic features, which affiliates eukaryotes in the phylogenetic realm with archaea. And here I've included a picture from the University of California Museum of paleontology. But that's just kind of an image that we have of one of the Lokis. So in order to support these theories, cultivation of the Asgardian archaea organism is necessary, because without a complete genome sequence, all these propositions are just assumptions. And this is where Dr. Hiroki Imachi and his colleagues come into play. They took on the endeavor of trying to cultivate, isolate, and analyze the needed strains. The hypothesis was along the lines of, like, if cultivation of the Asgardian archaeota strain is successful, then the previously accepted theories for evolution, such as the ones from Lynn, will not be supported. And the purpose of the study was to either confirm or reject the running theories of the Archaean derived hosts. That was the archaeal host cell involved in the endosymbiosis theory. And their approach was seemingly simple, but surprisingly long. Over the course of the next 12 years, Amachi's team would collect, separate, and analyze samples of uncultivated deep marine sediment microorganisms. They were able to collect by operating a methane-fed continuous flow bioreactor system for more than 2,000 days, and they were able to enrich and isolate the samples of the bioreactor community by inoculating them in glass tubes with simple substrates and basal media for approximately 7 years. They then took the cell turbidity formed and saw that it contained casamino acids, and they were able to compare these to clone library-based small subunit rRNAs, or 16S. So we have cultivation first. First, I'm going to kind of orient you guys to the graph that I have here. So along our y-axis, we are comparing the 16S rRNA gene, and along the x-axis, we have time in days. So the lines, I'm going to explain kind of what those mean. So the colored in circles are just casamino acids, and this is the basal that they're trying to grow this media in. The open circles are the casamino acids with 20 amino acids and powdered milk. The colored in squares is a 10-fold dilution of the open circles, and the open squares is a 100-fold dilution of the open squares. And then the colored in diamonds are just powdered milk, which is kind of used as the control group. So the result of this, static cultivation supplemented with 20 amino acids plus powdered milk resulted in stable growth at 20 degrees Celsius. And then, so once they were able to cultivate it, they found that 3 microorganisms were present in the sample, which you can kind of see on the pie diagram there and the B section of the graph. So there was Heliodisulfovibrio bacteria that made up the majority of the sample. And then the next largest sample was something that they termed MK-D1, which is the Archaean, the Asgardian Archaean strain. And then there was also a methanogenium archaeon, which is actually the symbiote of the MK-D1. So they found through metagenomic-based exploration and stable isotope probing experiments that the MK-D1 can catabolize 10 amino acids and peptides through centrophic growth with the Heliodisulfovibrio and the methanogenium through interspecies hydrogen transfers, which show that they found these 3 microorganisms together because they all relied on each other. And with fluorescent in situ hybridization, also known as a FISH technique, they were able to, by using the FISH and a scanning electron microscopy, they revealed a close physical association of the archaeon with other microorganisms. They then proposed the name Candidatus promethioarchaeum centrificum, but I'm going to call it MK-D1 because it's a lot easier to say. So genomic sequence of the MK-D1 reveals archaeal relationship to eukarya. So this is huge. This was literally the whole point of the research study. But by isolating the MK-D1, they were able to obtain a closed genome and construct ribosomal protein-based phylogenomic trees that actually show a clear phylogenetic sister relation between MK-D1 and eukarya. Therefore, MK-D1 represents the closest cultured archaeal relative of eukaryotes that we have today. So in the phylogenetic tree that I have included on the slide, it's kind of hard to see, but the red line indicates where MK-D1 is located, and it is actually the closest ancestor branch-wise we have to the domain of eukarya. The graph is based on 31 ribosomal proteins that are conserved like across all 3 domains. So they were able to like double check it with the other 2 domains as well. And even after doing that, it was still closer to Eukarya than it even was Archaea. And they also used about 14,024 sites of alignment of the tree construction, and it confirmed the presence of 80 eukaryotic signature proteins. So then we kind of jump into this E-cubed theory, or the entangle, engulf, indigenize theory. So this is the physical manifestation of this endosymbiosis, and it was probably independent of phagocytosis due to the proposed lack of sufficient machinery and energy, which they were able to see because of the electron microscope, microscopy. So alternatively, their theory was that the host archaeon engulfed the metabolic partner using extracellular structures and simultaneously formed a primitive chromosome surrounding structure that is topologically similar to the nuclear membrane. And to kind of orient you a little bit more onto what I mean by this, I have included this little video. We'll watch about a minute of it. It doesn't have volume, but it does have captions. Oop, gotta start it over. There we go. So these are the vesicles that they were able to see on the microscope, and this is what they believe the entanglement option of, or the entanglement portion of the theory comes from. And because they were symbiotic with each other, they proposed that instead of just being entangled with them, they would actually eventually engulf them, which is what's kind of being shown here. So I have included this picture straight from, it's one of the figures from their research paper. And so I'll kind of try to quickly break this down for you. But in A, we have the syntrophic fermentative host archaeon, which is suggested to degrade amino acids to short carbon fatty acid chains and H2. And then B, the host may have further interacted with a facultatively aerobic organotrophic partner that could have scavenged toxic oxygen, or O2, which is indicated in the red. And then C, the host external structures could have interacted with the aerobic partner to enhance physical interaction and then further engulf the partner for simultaneous development of endosymbiosis and a proactive nucleotide bonding membrane, which is in theory where the nuclear membrane came from. And then after engulfment, the host and symbiotes could have continued this interaction that's shown in B. And then the last one, we have endogenization of partner symbiosis by the host through delegation of catabolism and ATP generation to the endosymbiote and establishment of symbiotes to host ATP channel. So I've included this quotation. I'll read it for you guys real fast, but isolation and cultivation of MK-D1 brings us closer to understanding how eukaryotes may have emerged. However, it is important to emphasize the vast amount of time, roughly 2 billion years, that separates this modern day organism from the organism that evolved into the last eukaryotic common ancestor, also known as LECA, leaves many uncertainties. Although we can have reasonable assumptions on the events that may have occurred during the course of evolution, so the reason I included this quotation was to kind of highlight the importance to show that there will always need to be further research and data to either confirm or reject data presented in previous research, which I think is a kind of a nice segue into the next steps that Amachi wants people to take his research and do with. So, He said that given the phylogenetic relationship of MK-D1 with other Asgardian archaea and eukaryotes, estimating the physiological traits of the last Asgard archaea common ancestor is of the utmost importance. And further investigation into MK-D1 related Asgard archaea and more deep branching eukaryotes is, he says it's now required, and I completely agree with him, and could potentially provide valuable insights into the timing and progression of later lateral gene transfer and endosymbiote organogenesis towards the first mitochondrion and the formation of the endomembrane system altogether. So for the takeaway slide here that I have, I kind of just wanted to make sure you guys understood the origin of the eukaryotic life, how we have the endosymbiotic theory that was proposed by Lynn, and we kind of went through today how it wasn't accepted, and then it was accepted, and now they're kind of not accepting it anymore because of the recent research that has been done within the last decade. And then I also wanted to talk about how important it was that we were able to come up, and I say we like I was a part of it, but that scientists were able to come up with a way to cultivate this beforehand uncultivatable. I don't know the word I'm looking for, but they weren't able to do it, and now they are. And that's huge, because now that we can just do one strain, that means that we can do all of the potential strains that we can find in the methane seeps at the bottom of the ocean, which is where most of these extremophiles, or the organelles that are involved in the Archaea domain are found. So the genomic sequence indicated that the entangle, engulf, indigenize theory is much more likely than the original one. But I know it's kind of hard to understand, and I really also don't 100% understand it. I totally like, I want to push you guys, if you're at all interested in this, to look into it yourself, because it is revolutionary. And even though we don't have all the information on it, and it is still just a proposed diagram, I think it could be where evolutionary biology goes next. So if you're interested in that at all, definitely look into the E-squared theory. I want to give a quick shout out to Dr. Bobby for having put all of this together and allowing us to kind of do research on something that interests us. And I want to thank him for coming up with my very long or very lengthy title. I couldn't have done that, so thank you. And I also want to give a special thanks to Dr. Alford, who helped me understand the background information and introduced me into some of the themes that I brought up today. So the last slide I have here are my citations. And again, thank you for watching, guys.